Hello, welcome to our lecture on classification. This lecture is informed by the previous lecture, Lecture 2, which was our Introduction to Evolution. We're going to take the concepts that we learned about evolution and apply them to our groupings and characterization of the diversity of animal life. That's a system called classification, where we group animals together based on either morphology or phylogeny, depending on the system you're using. We're going to start with a quick overview about the history of classification and the system we are going to employ. The original, you can think of it, system of classification for the sciences was come up with by a guy named Linnaeus. Put an S on the end of that word and you've got his name. Now, the Linnaean system of classification is our traditional system, and we still use it to this day because it's so well established. It assigns every living thing, every organism, a position in a series of ranked categories. There are domains, which can be broken into a series of smaller groups called kingdoms. Kingdoms can be broken into smaller groups called phylum, or phyla, or phylum if it's singular. Phyla can be broken into classes, classes can be broken into orders, orders can be broken into families, families into genera, genus is the singular, and a single genus can be broken into many species. As you go down the ranks, you get more and more specific. Each subsequent rank has more requirements to fit into it, until finally you get to the species level, which is the most specific. Remember, a species is a single type of living thing. Now, the trick with the Linnaean system is that it was developed during a time where we had no knowledge of DNA or genetic relationships. So this entire system is based on morphology. What organisms look similar to each other? What organisms look different from each other? That's how we grouped animals in particular into their various phyla and classes all the way down to their species. So the key thing to remember about the Linnaean system is it's not informed by evolutionary relationships. This has a major failing, and that is, as you know now, genes can be activated or inactivated. Two organisms can be almost genetically identical, which definitely shows you they're related, but they can look very, very different from each other. If we used morphology alone, we'd be tempted to class them into different groups, even though they should be each other's closest relatives. That's the shortcoming of the Linnaean system. We are going to be using the phylogenetic system of classification to overcome this shortcoming. In the phylogenetic system, we group organisms based on their shared ancestry, because that's a reliable indicator of relationships. Here's an example of how the Linnaean system can be misleading. If you look at this diagram, down here on the bottom left, and you pretend that you can't see anything below this line, right? Pretend that you can't see what's in that blue box, essentially, and you're just looking at this group of organisms. You might be tempted to say that based on morphology alone, which is all you can see, that turtles, lizards, snakes, dinosaurs, and crocodiles all have a lot of morphological similarities. You might be tempted to group all of them together and say that they're each other's closest relatives. And that's exactly what Linnaeus did. He called that group the reptiles. Birds, however, look significantly different from the others. That led him to group them in a separate category called aves. Aves is just Latin for birds. But now, look below this line. Pretend the line's gone. Now you can see the actual evolutionary relationships between these different organisms. You can see that they all share a common ancestor way down here. Turtles are the oldest group, followed by all the descendants of this point. Now track all the way up until you get to birds. Notice that the closest relatives of birds on this tree are one of these groups of dinosaurs. Birds and dinosaurs are each other's closest relatives. In fact, other than the crocodiles, birds are the closest relatives of all the dinosaurs that ever lived. This is an indication that birds need to be included in whatever group you put dinosaurs. And since dinosaurs were the closest relatives of crocodiles, crocs need to be in that group as well. But lizards and snakes and turtles are all morphologically similar. They've got genetic characteristics in common with these groups, which really suggests that everybody on this tree needs to be grouped together. That indicates that Linnaeus' system of separating birds and reptiles isn't appropriate. They are one large related group. But if you look close at this tree, you'll notice it is possible to identify subgroups within this larger related set. Turtles kind of stand on their own out here. Okay? We could identify them as their own subset of this larger group, and then give them their own name, the testudines. Lizards and snakes are paired together over here. They're each other's closest relatives. They're what we call a sister group. Okay? We could group them together and give them their own name, the squamates or the squamata. Everybody else on this tree are all pretty equally distant from each other and equally close to one another. But they all descend from this ancestor, and nobody else on the tree does, except for the individuals in this particular box. It makes sense to give that large group its own name, the Archosauria. But then within that larger group, we have one subgroup that's the oldest, the crocodiles, which is part of a larger group called the Crocodilomorpha. It includes alligators, caiman, and gharials as well. This tree is a much more specific and evolutionarily informed version of classifying this particular set of individual species. This tree uses the phylogenetic system, informing our groupings by ancestry and evolutionary relationships. This is the system we are going to be using. Now, there are various terms that are used when discussing phylogenetics. So what's the difference between a phylogeny, an evolutionary tree, a phylogenetic tree, and a cladogram? Well, the answer is not very much. For our purposes, phylogenies, evolutionary trees, and phylogenetic trees will all show basically the same information, and they will include information on time. So they won't just show the relationships between groups. They will also show their respective ages and how old each group is, and whether or not each group is still around or if they've gone extinct. Cladograms are a little bit different. For our purposes, cladograms are going to show relationships only. No time. Cladograms will not show the ages of lineages, and they won't show whether or not groups are extinct. They will just indicate how closely they're related to each other. That's this point down here. Okay? What phylogenetic trees and cladograms both show are how different taxa are related to each other. Taxa is the plural form of the word taxon. A taxon is any named group of organisms. Reptiles is an example of a taxon, but so is eukaryotes. Eukaryotes are a taxon defined by the fact they all have cells with nuclei. So a taxon can be big, incorporating a lot of different organisms, or it can be very specific, like Homo sapiens, which is just humans. Sister groups are also depicted on both of our types of trees. Sister groups are each other's closest relatives on the tree. Outgroups are also present. You saw this in one of your previous labs. An outgroup is the most distantly related branch on a phylogenetic tree or a cladogram, and it gives context to the tree. It shows you where on the larger tree of life this group fits. It shows you what the ancestral characteristics of this group might have looked like. And it just gives you a sense of where this particular group fits into the larger picture of animal diversity. Finally, both of these tree types show you clades. Clades are a critical feature of phylogenetics, and it's really important you understand what they are. A clade is a group formed by an ancestor and all of its descendants. So a clade is a self-contained group formed by a single ancestor and all of its descendants. So let's take a look at this diagram here on the left. Anytime we have two branches that come together and meet at an intersection like this, we call that a node. And nodes represent common ancestors. So wherever you see a node like that, here, if we go to the right tree, here, 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 or here, all of those nodes represent an ancestor. Now you can think of the lines as being a summary of ancestors as well. Each of these lines represents hundreds, if not hundreds of thousands of generations of organisms that came before this point. Okay? But the nodes are a single obvious point of commonality between two separate lineages or lines. Both of these groups of organisms had to originate from this point. That is definitely a shared ancestor of theirs. Now, if we want to base classification on this, what we have to do is identify our common ancestor and then track all of its descendants, all the groups that evolved from it. In the case of the tree on the left, this node gave rise to just these two descendants. So to form a clade, based on this information, we would need to include both taxon A and B, and by default, the ancestor that they share. This is a self-contained clade, everything inside this box. Okay, that's your clade. Now, clades can be big or small. We can do the same thing going back one node on this tree, and as long as we include all of the descendants from that node, which would be A, B, and C, and we include that ancestor, we have made another clade. Looks like that. Okay. So let's practice this on the tree on the right here. Okay. So take a look at that yellow box. Does this yellow box represent a clade? Let's check. We'll find the oldest ancestor in the box, which is this one, and we'll trace it forward. We've got one descendant here, one descendant here, and those are the only two descendants from this ancestor. They are both included in the box, so is the ancestor. There's no weird extra descendant lineages over here. There's no ancestors back here that are left out. This is a self-contained clade. So that yellow box, definitely a clade. Now what about the blue box? We're going to repeat that same process. We have an ancestor in the blue box here, but it's not the oldest one. The oldest one is back here. If we trace the descendants from that ancestor, we have 3 of them. all three are inside our box, meaning, and there's no extra descendants that are left out over here. There's no additional ancestors that we've reached back and included as an extra ancestor. We didn't do that. If it's just these three descendants, and they all trace back to that one ancestor, it's a clade. The blue box is also a clade. Notice you can have smaller clades within larger ones. That's totally fine. All right, take a look at that red box. You tell me, is that a clade? Well, we've got our oldest ancestor here. Trace out all of its descendants. Make sure they're all inside of this box and there are no extra descendants. And we didn't try to force in some random extra ancestor. As long as there are no extra descendants, no extra ancestors, everybody's in the box, it's a clade. What about the green box? Yeah, what do you think? Well, we've got an ancestor here in the green box, and here, they're all included, but the oldest ancestor is way back here. Starting with the oldest ancestor, we'll trace forward to make sure all its descendants are included. There's 1. There's 2. There's 3, 4, and 5. Those are the 5 descendants of this ancestor. Check and see if they're all inside this green box. They are. There are no extra lineages or lines that we left out. There's no extra ancestors we tried to include. As long as everybody is included inside the box, which they are, it's a clade. Clades are a critical component of phylogenetic classification. We always discuss this system in terms of clades. So here's a little bit more practice. Green box here, definitely a clade. Got our oldest ancestor and all of its descendants included. Okay. Now over here, this blue box is actually a clade as well. This is a weird kind of clade. Okay? Remember how I said these lines are basically representations of hundreds or thousands of ancestors that came before this point where these two groups split? What that means is if you're looking at a particular group of organisms that are at the tip of one of these branches, the line that comes before them represents hundreds of millions of their ancestors. So this entire line is made up of ancestors that only belong to this one group. So is it a clade? A clade is an ancestor and all of its descendants. Any point along this line counts as an ancestor. Okay? If you move forward from any of those points, the only descendant group you get is the one at the end of the branch. So each single branch counts as a clade. Now, one of the reasons that Linnaeus' system of classification is so troublesome for us now is because groups exist within his system that are not clades. For example, let's look at this tree on the bottom right first. And before we actually talk about it, I want to teach you one more term. That term is lineage. A lineage on a phylogenetic tree is represented by these lines. So each of these lines is a lineage. And it's not just the line that ends in the tip of our branch on this tree. A lineage is this part of the line plus this part plus this part, tracing back to the root of the tree. A lineage is a descendant plus all its ancestors. So each of these branch tips has a lineage, a line that comes before it. Think linear as in line. It's its line of descent. All the ancestors that came before that gave rise to that particular descendant group is its lineage. Okay. So I'm going to start referring to these branches as lineages instead of lines. Okay. All right. Look, now let's go and look at this tree on the bottom right. Okay. This orange box represents a clade made up of these 5 lineages. Okay. If we trace them all back to their oldest shared ancestor, the oldest node is here, we find that that ancestor's descendants are this entire group on the left, plus this entire group on the right. So far so good, but there's one more lineage out here, and it's left out. It's excluded. Remember, a clade is an ancestor plus all of its descendants. Not all its descendants are included inside of this orange box, which means this orange box cannot be a clade. Let's look at the tree on the left, red tree. It's made up of these 3 lineages and this ancestor. Right? So find the oldest ancestor in the box. It's here. Trace out its descendants, 1, 2. Okay, so far so good. All of its descendants are in the box. It's in the box. But then we have this extra lineage. Uh-oh. We've got a bonus lineage, an extra one. We need to connect it to our other 2 lineages in order for this to be a clade. In order to do that, though, we've got to backtrack through 1, 2, 3 ancestors that are not included in our box before we would actually get to this lineage. We've got too many lineages here. We've left out a bunch of ancestors. So we don't just have one ancestor in the box, we've got 1, 2, 3 ancestors in addition to our one ancestor that's included. Right? We have to cut through too many ancestors to try to connect this extra lineage. Right? We've added extra ancestors as we have added this extra lineage. It's not a clade. There's too many lineages here, too few lineages on the right. Okay. But groups like this are very common in the Linnaean system. Go back to our first slide. Check out our entire tree here on the left, the reptiles tree. Let me erase all the notes, give you a clear view. This should look pretty familiar from that slide we just examined. We've got a group of lineages that all connect to a common ancestor, but not all of its descendants are included. One of its descendants are left out. The reptiles aren't a clade because the birds are excluded. Since groups like this are so common with the Linnaean system, we actually give them names. Groups that are true clades, that count as clades, are called monophyletic groups. A monophyletic group is the same as a clade. Okay? These are synonyms. Okay? Clade and monophyletic group mean the same thing. Paraphyletic groups are partial groups. It means they are missing a lineage. They have left a lineage out, making them partial or incomplete. That's reptiles. Polyphyletic groups have too many. Think of poly as many. They've got too many lineages. They've got extra lineages that have been shoved in that aren't actually closely related to everybody else in the group. We're going to encounter a lot of polyphyletic and paraphyletic groups as we move forward in the semester. Now, what we've been looking at up till now are actually cladograms. None of those trees have incorporated time. If we start putting a time axis onto our trees, like you see here, what we've created now for our purposes is a phylogenetic tree. Time is represented by the length of each branch. Long branches represent old lineages, ones that have been around for a long time. We've got fossils of jawless fish that go back to about 495 million years ago. Marsupials and placentals, two different types of mammals, are relatively young. Our first fossils of them and our genetic evidence date back to about 65 million years ago, give or take. What you also see on these trees are branches that are short. Modern time, today, is always this topmost line of the tree. Any branches that don't make it to that topmost line represent groups that are extinct lineages. And you can see, based on the time axis, approximately when they died out. So even if these branches weren't red, you'd know that they represented extinct groups because they literally don't make it to today. Now, why are we so hung up on the phylogenetic system as opposed to the Linnaean system? Well, other than it showing more accurate relationships between lineages, because it's grouping organisms by their ancestral relatedness, so true genetic relationships, it also fixes some other issues with the Linnaean system. The Linnaean system is very rigid. Every organism on the planet belongs to a domain, a specific kingdom within that domain, a specific phylum within that kingdom, all the way down to species. We call these ranks, and just ranking organisms into the same sorts of categories, it insinuates that different ranks are probably similar to each other, that taxonomic levels are equal across all groups, that if you're looking at a family of plants, any given plant family probably has similar diversity to any other plant family. And any plant family probably has similar diversity to any animal family. Now let's test to see if that's true. If you'd expect to see a similar number of genera within a plant family as you would within an animal family. We're going to compare the animal family Felidae, which are cats, to the plant family Orchidaceae, which are orchids. Okay. These are both families, right? They have the same taxonomic rank. You'd probably expect them to have similar numbers of genera, similar numbers of species within them, similar diversities, because they're the same rank. But is that actually true? When you start comparing the numbers, here's what you see. There are about 20,000 individual orchid species. 20,000 species within family Orchidaceae. Within family Felidae, there are 35 species of cat. Clearly, these two families don't have the same diversity. The felids are much, much less diverse. Okay, well, maybe it's not diversity that makes families similar to each other. Maybe they're the same age. Maybe all families evolved about the same amount of time ago, about the same point in history. All right, well, let's test that. The first ancestral cat, probably lived about 30 million years ago, MYA, for millions of years ago. While the first orchids, they lived about 100 million years ago. Okay, so that's not the similarity either. Orchids are significantly more diverse and they're older. All right, well, maybe they have similar levels of differentiation. Maybe all of the types of cat are as related to each other as all the types of orchid are related to each other. Maybe all cats are first cousins and all orchids are first cousins. And maybe it's relatedness within each rank that defines the rank. Well, one way to test that is to look at the biological definition of species, which has to do with reproduction. Can two different genera of orchids reproduce successfully? Can two different genera of cat reproduce successfully? That'd be one way to test this relatedness hypothesis. Well, if we take two example genera of orchids, Laelia and Cattleya, we find that they can hybridize. They can reproduce. Sweet, that works. They can reproduce. Let's test that with cats. Let's take genus Felis, which is what domestic house cats belong to, and genus Panthera, which is what big predatory cats like lions belong to, and see if they can reproduce. Nope, they can't. They can't. They are reproductively incompatible. They're isolated from each other. So this relatedness hypothesis falls through as well. So what actually defines a family? Nothing. Morphological similarity. That's what defines a family. And you know now that morphological similarity is very misleading. This is why the Linnaean system is so problematic. The phylogenetic system is more complex, but it's more reliable too, and it's more informative. What we're going to do now is talk about a field of study called cladistics. Cladistics is where you take the tools of phylogenetics and you apply them to help hypothesize relationships between organisms, both within clades that you've already defined and between clades, to get a sense of how evolution has affected larger and larger scales of group. Now you can do this by comparing characteristics as they've changed over time. Now this can be dangerous because characteristics are morphology. But remember, morphology is defined by genetics, by genes. As long as you are taking genetics into account and you are mindful of the fact that simple morphology can be unreliable, you can make informed choices about what characteristics can be reliably used to reflect ancestry and shared genetics. But you have to be careful about what characteristics you use to define relationships between groups. And we'll talk about that in a few slides, how you make that choice. Now here's how you categorize characteristics when you start to use them to define these cladistic relationships. There's two big categories of characteristic. There are ancestral characteristics, which are the old versions of characteristics that showed up early in the ancestry of a group. And then there are what we call derived characters. These are characteristics that have changed over evolutionary time as different groups have speciated and become more specialized to their environments. Because it's science and we have to ascribe our own terms to everything, ancestral characteristics are called plesiomorphic characteristics. Plesiomorphic. Derived characteristics or new characteristics are called apomorphic characteristics. By default, if a apomorphy does not show up for a particular lineage, they have the plesiomorphic version of a character. Here's what that looks like. We're going to revisit our reptiles down here. If you go back to the oldest ancestor on this tree, you'll find that it has the plesiomorphic state for this clade, meaning the plesiomorphic set of characters. Those characters are an absence of feathers and an absence of wings. So not having something is just as descriptive as saying that you have it. No feathers and no wings, those are physical characteristics, just like having feathers or having wings would be. The plesiomorphic state for this group is to not have feathers or wings. That's true for crocodiles. It's true for all of our dinosaurs. But it's not true for our birds. Our birds are different. Birds evolved both wings and feathers. They changed. Their physical covering no longer looks like the rest of these descendant lineages. They have the apomorphic version of these traits. They have feathers and they have and they have wings. Okay? They changed right about here. Okay? That's the apomorphy. Now there are two types of apomorphies that you see. If you're looking at a large tree, like the tree for the primates over here, what you'll start to see is that there are some apomorphies that show up early in the evolutionary history of a group, and then they're passed down to multiple different lineages. A lot of different groups inherit them, especially if they're really, really beneficial adaptations. These apomorphies that show up early in the history of a group and then are passed down to multiple descendants, they're called synapomorphies. Synapomorphies are shared. Think S for synapa, S for shared. Okay? And they're shared across several related lineages, and that's key, is the lineages must be related. They've got to be within the same tree. So, for example, I'll show you an example on the next slide. First, let's look at the other kind of apomorphy. The next kind of apomorphy is what's called an autapomorphy. Okay? Autapomorphies are derived characters. Okay? It's an apomorphy after all, but they're unique to only one lineage on your tree, just one. These are a defining characteristic of that particular lineage. Okay? So let's take a look at this on a larger version of this primate tree. All right, let's look back here at our ancestral primate. Okay? This ancestral primate had 5 fingers. Now think about this. That's 5 fingers, not a thumb. So if this is a hand with a thumb, 1, 2, 3, 4 fingers, that is not what these guys had. They had literally 5 fingers. Like that. All the fingers are in line with each other. They don't have a thumb that can wrap around and face the fingers. Instead, all they can do is curl their fingers forwards like this. When you have a thumb, you can curl your fingers forward and curl your thumb inward, allowing you to grip things more easily. But the ancestral state for all primates is to have 5 fingers. These old lineages of primate, lorises, plateaus and lemurs, the tarsiers, they have 5 fingers still. Okay? And they're very interesting looking. I suggest you go and Google them. They're really neat animals with very interesting hand morphology. But at some point here, on this descendant lineage, that changed. Right about here is where having a thumb showed up. So now, descendants from this point, descendants from really this ancestor, they have 4 fingers plus 1 thumb. You see that in New World monkeys. I'm just going to write the number of fingers, 4, and then the thumb, plus 1. You see that in your Old World monkeys, 4 plus 1. Gibbons, orangutans, gorillas, all of these guys are 4 plus 1. 4 fingers plus 1 thumb. And that includes us too. We all share this synapomorphy of having 4 fingers and a thumb. It's shared among us. It's not unique to any of these groups, but it's a change from the ancestral state for the group. So it's definitely an apomorphy. What about autapomorphies? There's one obvious one on this tree, and it belongs to us. We have an extra large brain compared to the size of our bodies. It's out of scale with the rest of our bodies. That extra large brain also has extra folding on its surface, which helps increase our processing power. It's one of the reasons why we seem to have so much more intelligence and problem-solving skills than most primates. Okay? But that's unique to us, which makes it an autapomorphy. All right, so we've said that there are two different types of apomorphies. But are there multiple types of plesiomorphies? Do symplesiomorphies exist? Absolutely. Absolutely. There are plenty of shared plesiomorphies. For example, the ancestral primate also had fur. This is a really easy one. Almost every one of these groups has fur. People argue about whether humans do or not. I argue that we have derived fur in the form of our head hair and our body hair, so I'm going to draw on a line there. All of these groups share that characteristic of having fur. Okay? We all have it. That makes fur a symplesiomorphy. Okay, so you can have shared plesiomorphies just like you can have shared apomorphies. When you can identify characteristics that are reliable indicators of common ancestry, you can actually use them to build your cladograms. Okay? But what are these reliable indicators of common ancestry? What kind of characteristics make the cut? The best characteristics to use as information for building a cladogram are called homologous characters. Homo meaning same. That word same in this case refers to the fact that they're being inherited from the same ancestor or a common ancestor. These are characteristics that indicate shared ancestry because they could only have been inherited from a shared ancestor. For example, tetrapody. Tetrapody is the state of having 4 limbs. Limbs do not have to be arms and legs. They can be arms, legs, or wings. Wings count as well, okay? If you look at a tree of the tetrapods, you'll find that they share an ancestor right back here. Some point just before that ancestor started to split off into multiple groups, having 4 limbs evolved. That trait of having 4 limbs was passed down to every single one of these descendant lineages. It's an indication that all of our modern day tetrapods likely inherited that state of being a tetrapod from this ancestor. Notice it's the oldest ancestor for the group. The simplest explanation for why they all have that characteristic is that they got it from that common ancestor, as opposed to each group developing it somehow individually, and just by pure luck, they all developed the same character of having 4 limbs. That's really unlikely, having it evolve 1, 2, 3, 4, 5, 6, 7 separate times. It's a lot more likely it evolved once, and then it was passed down. This is what we mean by a reliable indicator of shared ancestry. Tetrapods all share this trait. They all trace to one ancestor. It's very likely that ancestor is the individual who gave rise to this character in this group. Now, there are times where characteristics, as always, can mislead you. Remember, morphology can lie. There are characteristics that look very similar to each other, almost identical, but have separate evolutionary origins. They came from different ancestors. We call these homoplasies. They're also sometimes called analogous characteristics. Remember, an analogy compares two dissimilar things to point out what they have in common. So if I'm saying that a woman's hair was as golden as the sun, I'm not saying her head is covered with a fiery ball of nuclear plasma. I'm not saying her hair is the sun. It's an analogy, a descriptive one, but they are not the same thing. Analogous characters work the same way. I'm going to call them homoplasies because that's the more precise term. Homoplasies are characters that appear very similar, but they've got separate evolutionary origins. Another way to put this is that they are coded for by different genes. And if they're created by different genes, it means they didn't evolve from the same place. They got two different genetic origins. Characteristics like this evolve in different groups, completely separate from each other, because of what's called convergent evolution. Convergent evolution happens when a certain characteristic is so good at helping an organism survive or reproduce that every time it shows up in the evolutionary history of a group, every time it pops up in a new lineage, it's selected for and it becomes common. So wings are a great example. Okay? Wings exist in bats and in birds. But bats and birds are very distantly related to each other. They're separated by millions of years of evolution. They're separated by many, many branches on our phylogenetic tree. Bats and birds did not inherit wings from somewhere back here. If they had, it would mean literally every other species of mammal, every mammal, and many reptiles, not every reptile, but many reptiles, would have to lose their wings in order to explain why bats are the only mammal that has wings now and why birds are the only reptile that have wings now. Does it make more sense for thousands of other groups of organisms to lose their wings in evolutionary reversals? Or does it make more sense that wings just happen to evolve twice, once in bats and once in birds? The simpler explanation is that wings evolved in vertebrates during two separate events. Wings have actually evolved 4 different times. This is 2 of the 4 times. So I'll put little stars here to make this make sense. These are examples, 2 examples of the 4 times wings evolved in animals. The other 2 times were in insects, and then again in a group called the pterosaurs, which were the winged dinosaurs, usually referred to as pterodactyls. Now look at the structure of bat and bird wings. Once you start looking at them closely, you'll start to see the differences. They become apparent. Now you can tell these are both descendants of a vertebrate, a tetrapod vertebrate, because they have similar bone structure. The bones they inherited from a common ancestor, but the arrangement of the bones is what changed. They both have a humerus up here, an upper arm bone, okay? They both have a radius and an ulna down here, connected to the humerus in approximately the same spot. They both have wrist bones, carpals, but there's different numbers of them. They both have mid-finger bones as well in here. Okay. Those are your metacarpals. But then they also have their phalanges, the finger bones out here. But notice again, they have different numbers of them, different arrangement of them. This is where the differences really show up. Remember that those bone positions have to be coded for by genes. You've got different genes coding for different bone positions in these 2 animals. It indicates that these are not the same structure. They were not inherited from the same ancestor. They just have to do a similar job. Keep the animal in the air. We call any structure that does that a wing, even though physically they are so very different from each other. Now, how are you going to use these homologies to actually build a tree? This is where things get kind of fun. First thing to know is that you've got three major categories of homologies that you can use. You can use evidence of the same basic structures, meaning the same bones present in each group, even if they're structurally modified a little bit differently from each other. It's still an indication that that basic bone structure was inherited from a common ancestor. You can look for the relationships between physical features. If we stick with the bone analogy, that would be joints. This on the left is an X-ray of a lizard hip. On the right, X-ray of a dog hip. Two very different animals. They walk in very different ways. Yet look at how similar the connections between the joints or the connections between the bones are. Same relative positioning, same relative connection. Okay. You can also look at the development of structures over the course of an animal's lifetime, starting with their embryonic development. This is an alligator embryo here on the left, a mouse embryo on the right, two very different organisms. But the precursor structures to their limbs, their arms and legs, what we call limb buds, they show up at approximately the same time in development for both groups. So if structures arise at about the same time during development in two vastly different organisms, it could be that literally the timing of their development is what's been inherited from an ancestor that they shared. All right, just a quick review of how we term our characteristics. Remember that plesiomorphies are ancestral versions of a trait depicted as gray dots here. So these nodes are gray, indicating these ancestors had this version of the trait. These descendants also have the gray dot, indicating that they inherited this ancestral trait, this plesiomorphy. Black dots here indicating that this is a different form of the trait, making these two lineages the recipients of the apomorphy, a shared derived characteristic. Okay. But remember, just because these two characters look the same, it doesn't mean that they are from the same ancestor. They could have evolved totally separately, making them homoplasies. But how would we know? We could do some genetic testing. We could do some pretty rigorous examination of the fossil record and try to determine what this ancestor looked like. If we find that the ancestor had the same version of the trait that our descendant lineages do, that's a safe bet then that this is a synapomorphy, and our two descendant lineages inherited that characteristic from the ancestor. Okay. Remember that even if just one lineage exhibits the change in the trait, it's an apomorphy, and in this case it makes it a unique apomorphy or an autapomorphy, when only one lineage has it and it defines them. All right, so what's this? We've got the apomorphy here, we've got the apomorphy over here. So what kind of derived trait is that? Is that an autapomorphy? Is it a synapomorphy? Plesiomorphy? It's another example of a homoplasy. This is a definite homoplasy. There's no way around it. This could be birds and insects. They both have wings, but they definitely did not inherit those wings from the same ancestor. Too many lineages in between them would have had to lose the wing in order to explain them both having it. And what this tree is even telling us is that the plesiomorphy, which in this case would be no wings, that absence of wings is what's being passed to these groups. So this tree is very convenient because it's telling us that these ancestors did not have wings. Only these two descendants do. It means it has to be a homoplasy. This is called a character matrix. It's a chart that has a series of homologous characteristics that you can use to build a phylogeny showing the relationships between the depicted lineages over here. Okay. So what we are going to do over the course of the next couple slides is use these characteristics and our understanding of phylogenies to build a tree that depicts the relationships between these groups of specifically vertebrates, organisms that have a bony vertebral column, or I'm sorry, just a vertebral column. Doesn't have to be bony. Here's how you do this. I like to start by looking for characteristics that split our organisms into two groups of roughly the same number. Okay. So amniotic egg is a good characteristic for this. Half of our lineages don't have it, and the other half do. So we can actually start building our tree using just this information alone. Here's how. Notice that the ancestor is listed here at the bottom. Even if the ancestor was not provided, you could deduce what its characteristics would be. Character matrices always have binary options, meaning your lineage either has the character or it doesn't. The ancestor, by default, is said to have none of the characteristics, because what makes these characters interesting is that they're all derived. These are all apomorphies. The plesiomorphic state for this group is to not have any of these characters. And since the ancestor has all the plesiomorphic versions of the traits, it doesn't have any of these new derived versions. So, which of these groups is going to be closer to the ancestor, closer to the root of the tree? Well, it'll be the ones that have more in common with the ancestor. The ones that don't have any of these fancy new apomorphies. So we can draw a basic version of our tree based on that information alone. These three lineages, sharks, ray-finned fish, and amphibians, all lack the amniotic egg. Primates, rodents and rabbits, crocodiles, and dinosaurs and birds all have the amniotic egg. By mapping the trait here on the tree, we show that this is the character that split these two sets of lineages apart. Those that lack the trait go before the line, closer to the ancestor, who's down here. Those that have the trait go above the line. Okay. We're going to continue to do this now to tease out the relationships between these two sets of lineages. So I'm going to go ahead and redraw this rough tree on the next slide. So here you can see a little miniature version of our character matrix. I'm going to go ahead and draw in the backbone of our tree. And I'm just going to copy what we had on this previous slide. So we've got a cluster of 4 lineages up top. Now notice how I'm drawing this, how careful I'm being to make sure that all of these lines meet at the same place up here. All of the lineages we're working with still exist today. So I want to make sure that all of these lines reach all the way up to this dotted line so that it's very clear that there are no extinct lineages on this tree. This is an expectation. I expect your trees to look like this. Okay, so if I ask you to draw one of these trees on an exam, and I'll give you a hint, I'm going to ask you to draw one of these trees on an exam. My expectation is that all of your lines will match up at the same level if all of your lineages are still around, if they're all still alive. All right, so we've got 3 lineages here, 4 up above, and they are separated by this apomorphy, the amniotic egg. The other thing I want to point out is, look at the end of the tree, this last branch. That final branch has to have a lineage on it. It cannot be blank. You can never have this final branch just hanging out empty. That's a lineage. Something has to be written above it. In this case, what I am going to put there are, based on the previous slide, are dinosaurs and birds. They're going to occupy that last branch. So I'll abbreviate to D and B. Then we have crocodiles and their relatives. We have rodents and rabbits. And we have primates. Then these last three lines are amphibians, ray-finned fish, RFF, and then sharks and their relatives. Okay, let's start by working on our lower group of three. Let's see if we can tease these three lineages apart. So keep in mind now, on our character matrix, we are only interested in these top three groups. So everybody above this line. I draw it in because it helps me focus. All right. Now to tease apart these three, I am going to look for the lineage that is most similar to the ancestor, who would be located down here. Okay? Remember, the ancestor lacks all of these characters. So we're looking for the lineage that has the lowest number of these apomorphic characters. That's going to be the sharks and their relatives. Out of all these apomorphies, the only one they have are vertebra. They're going to be the closest to the ancestor out of all of our lineages. So that means that I can erase the line for sharks and redraw it like so, because I am confident they are the ones who have to be closest to the ancestor. But they are different from our ancestral group. Right? Ancestors don't have vertebra. Sharks and relatives do. That means we need to write in that the apomorphy of having vertebra evolved at some point after the ancestor, but before sharks. All right, now let's see if we can tease apart ray-finned fish and amphibians. So are there characteristics where one of these groups has the character and the other doesn't? Well, they both have vertebra, that's not helpful. They both have a bony skeleton, also not helpful. What about four limbs? Yep. They both have yep, as in only one group has four limbs. They mismatch. Bony skeleton is present in these two groups, but not in sharks. So we just discovered two things. One, bony skeletons evolved here, after sharks, but before our other two groups. And notice that everybody after this point on the tree also has a bony skeleton. It logically fits here. We've also discovered that having four limbs, being a tetrapod, is present in amphibians but not in ray-finned fish. Not having four limbs is an ancestral trait. It's a plesiomorphy. So the group that lacks four limbs, our fish, are going to be closer to the ancestor than the group that has four limbs, the amphibians. So I'm going to erase the amphibians line and draw it further up the tree, showing that ray-finned fish diverged before amphibians, and sometime in between them, the character of being a tetrapod, having four limbs, that evolved. The bottom of our tree is done. We have determined that every one of our lineages has vertebra, okay? So we don't have to worry about vertebra anymore. We've determined that every lineage except for sharks has a bony skeleton, so we don't have to worry about bony skeletons anymore. We've determined that every lineage except for our fish and our sharks are tetrapods, so we don't have to worry about that character anymore either. And everything above this blue line, we have mapped onto the tree. So what this leaves us with are just these remaining 4 lineages and these last 3 characters. But remember, we used amniotic egg to split our lineages into 2 groups way back at the beginning. So amniotic egg we already have used as well. That only leaves us these last 2 characters, presence or absence of hair, and presence or absence of 2 post-orbital fenestra. Now that's kind of a mouthful. Imagine that this is a skull, okay, seen from the side. There you go. Post-orbital fenestra. Fenestra is a hole. Post-orbital refers to the orbit of the eye, the hole in your skull where your eyeball sits. Some groups of vertebrates have openings in their skull that sit behind the eye, hence the word post. Post-orbital means behind the eye. You're going to see these when we study the vertebrate groups that have them. And I don't want to give it away yet, which ones those are. Right, so these are our 2 remaining apomorphic characters. So do either of these characters help split up our last 4 lineages? Well, yeah. 2 of our lineages have hair and 2 don't. But if you look at the second characteristic, we have the same relationship. 2 of our lineages lack post-orbital fenestra and 2 have it. So how does this work? Well, look at the relationships. Crocodiles and relatives and dinosaurs and birds both have this characteristic and they both lack this one. It indicates that they are more closely related to each other than either is to our 2 groups above, rodents and rabbits or primates. So what I can say with confidence is that crocodiles are most closely related to dinosaurs and birds. These 2 lineages are going to wind up next to each other on this tree as sister taxa. If I look at then rodents and rabbits, I see that those 2 lineages also have this in common. Only in this case, they both have hair and they both lack post-orbital fenestra. They are each other's closest relatives on this tree as well. So how do I represent that? Well, I could draw their lines like this, but that doesn't indicate that close relationship. It also makes it difficult to know where to put their apomorphy, which is hair. If dinosaurs and birds are up there at the end of the tree, I can put 2 post-orbital fenestra right before them on our backbone line. Indicating that both of these lineages have it, but no one before does. But then where do I put hair? If I put hair down here, before primates, it implies that every group after this point has hair. But we know from our character matrix that crocodiles and dinosaurs and birds don't have hair. So if I place hair there, I'd have to put loss of hair here. Now that's 2 changes to our evolutionary history. Is there a simpler explanation for this? Well, what if instead of saying that hair evolved and then was lost, what if instead we say that primates, rabbits, and rodents are all descended from a common ancestor that evolved after this group branched off from the main tree? If I draw it like that, now I can say that hair just evolved in this one ancestor that they share. It evolved once and then was passed down to these two lineages. This is the smaller number of changes that still explain the pattern you see. You always want to explain your evolutionary trends with as few changes as possible, as few instances of new traits evolving or ones being lost, because that is the simplest explanation. It's a lot less likely that a trait evolved once and then was lost and then evolved again and then was lost, as opposed to it just evolving once and then being passed down from that ancestor to many lineages. This simplest explanation strategy is referred to as parsimony. Parsimony is using the relationships on this tree, using these characters to explain the relationships on the tree in as few changes as possible. So if you find that when you're building a tree, you're having to say that a trait evolved in one group and then was lost in another and then evolved again in a third in order to explain which characteristics are present in which groups, it means that you're probably using too many changes. And there's a simpler way to draw your tree. The last thing that I want to talk about is how we reconcile the Linnaean taxonomic system with modern-day phylogenetics. Here's how I want you to think about these Linnaean taxonomic rankings in a way that makes sense phylogenetically. Think of each subsequent taxonomic level as you move down from domain to kingdom to phylum, on and on. Think of each new level as having a more and more specific set of apomorphies that an organism must have in order to fit in that new rank than in the level before. Here's an example. Let's talk about Panthera pardus, the leopard. This panther is in domain Eukarya, and to be in that domain, all you have to be is eukaryotic. That's it. One apomorphy in order to belong to this domain. But panthers are in kingdom Animalia as well. There are 4 kingdoms within domain Eukarya. Only one of them is kingdom Animalia, and to fit in this kingdom, you must be eukaryotic still, because you're part of domain Eukarya, but also heterotrophic and multicellular. Hetero means other, trophic means eating. Means you've got to eat other organisms, such as plants or other animals, in order to survive. Within kingdom Animalia, there are a large number of different phyla. Leopards are in phylum Chordata, which is right here. If we focus in on that phylum, we'll see to belong to it, you still have to be eukaryotic, heterotrophic, and multicellular, because this phylum is within kingdom Animalia, which is within domain Eukarya. But to belong to this particular phylum, you have to have this structure called a notochord. It's a rod that runs down your back to provide support. There's a number of classes within this phylum. Leopards specifically are in class Mammalia. To belong to this class, you've got to be eukaryotic, heterotrophic, multicellular, and have a notochord. Because again, this class is part of all the preceding ranks that came before. But now you also have to have hair, and you have to be endothermic, meaning you generate your own body heat using your metabolism. We'll talk about how that works in a later lecture. You see how this gets more and more restrictive as you go down? Leopards are in order Carnivora. In order to be in this order, you have to have all the characteristics that have come before and have canine teeth, the pointed teeth in the front of your mouth, and you've got to eat meat, other animals. Leopards are also in family Felidae within order Carnivora. To be in this family, you've got to have all the characteristics that came before, and also these, claws and whiskers. Within family Felidae, these leopards are within genus Panthera. To be in this genus, you have to have every characteristic that came before, all of these. plus whiskers, spotted or striped fur, and large feet. But to be within species Panthera pardus, species name is pardus, you have to have all of these preceding apomorphies plus specific autapomorphies. And those autapomorphies in this case tend to be thickness of fur and the shapes of these spots. So think about the accumulation of apomorphies as defining each rank as you get more and more specific. There's one more topic we have to cover. It's how phylogenetics loops back around and relates to evolution, specifically something called macroevolution. What we discussed in the evolution lecture was microevolution. It's evolutionary change at the scale of a single population, just a single group of related organisms. The mechanisms that cause it are things like migration and mutation, which introduce variation, and then genetic drift and natural selection, which affect the characteristics of the population over generational time. Macroevolution is larger scale. It looks at big picture trends and patterns in the diversification of all living things. We're looking at the evolution of entire groups, not just a single species. So instead of looking at the evolution of just leopards, we'd look at how all big cats had evolved, or how all cats of any sort had evolved. Macroevolution examines things like the appearance of new lineages, that would be speciation, how stable lineages are. So remember those lineages that are really old, like jawless fish? Why are they that stable? How many of them are there that are that stable? We look at big patterns of evolutionary change over the course of the history of life, and we look at what species are still extant and which ones have gone extinct, and under what evolutionary or environmental conditions. You can see macroevolution reflected in phylogenies. The longer lines on a phylogenetic tree indicate lineages that have been around for longer. Lines that terminate before they hit the top margin of the tree are lineages that have gone extinct. You can also draw phylogenies that describe how certain characteristics have changed in groups over evolutionary time. Usually you're looking for one of two trends. They're called stasis and character change. So if you look at this phylogeny on the left, body form is what's being depicted, either smaller body forms here or larger ones here. Time is on the y-axis. And what you see is that there's a speciation event, and then this lineage remains very stable and small in size, while this one remains very stable and large in size. After that speciation event, these lineages remain static in their body form, meaning unchanging. Now look at this chart on the right. After our speciation event, this lineage on the left gets smaller and then bigger and then smaller, a little bit bigger, kind of messes around in here, smaller, much bigger, and then sort of drifts. The one on the right wiggles around a little bit too, but progressively gets bigger and bigger as time goes on. Neither of these lineages are static. Both of them are showing a lot of changes in the state of that character over time. That's referred to as character change. And oftentimes, this sort of character change happens because of environmental conditions changing over and over and affecting the evolutionary conditions faced by that population or that lineage. And that's it. Thanks for watching.